First, I would like to thank the organizers for inviting me to have this talk dealing with lake sedimentary DNA. As you have seen from the title, the challenge is here for us to reconstruct long-term dynamic of lake biodiversity. But in our research group, we are more particularly interested by plankton communities and microbial communities. I am a limnologist actually, and one issue that led me to start with paleoecological approaches some years ago is the question of timescale. We know that anthropogenic pressure can threaten lakes, as for instance eutrophication and climate change, with ecological trajectories occurring at decadal or centennial scale. But yet it remains unclear the extent to which lake biodiversity has changed during the Anthropocene, and this is true in particular for microbes. There is clearly a lack of adequate long-term data related to biodiversity. Another issue is related to reference conditions. We need, of course, to define baseline conditions in order to see how far the system has diverged from its baseline, but we certainly need also to consider historical range of variability for our ecological metrics, keeping in mind the potential legacy effects, meaning the ecological inheritance that could influence current communities and the vulnerability of the lake to new drivers. So understanding how history constrained community resilience is of course critical. Well, it seems that sedimentary archives offer one of the best opportunities to tackle these two issues. Indeed, these natural archives simultaneously capture information from past environmental conditions, as well as species, population, communities with various proxies. And this allows, of course, to explore pre-perturbation states, timing of change, and the potential resiliency of the system. If we come to DNA, of course, DNA can complement traditional proxies as pigment macrofossils with potentialities to investigate various levels of past biodiversity. And we can use this DNA-based method either to analyze isolated cysts, so targeting individual within a population, for example, with an FEP of cladoceran, as illustrated here, or we can try to get an access to a large diversity of biological component from the analysis of what I have called here bulk DNA. So here the interest in this case, and I will focus more particularly on bulk DNA in this talk. So here the interest is that DNA extracted from few grams of sediment has the potential to reveal some information from DNA originated from all taxa that once lived in the lake. Of course, this means that we could have some communities from benthic zones or pelagic zones, and since some bacteria in archaea can live and be active in sediment, some study may require to discriminate DNA associated with these living cells, or may require also, of course, an authentication of ancient DNA signal. So the application of sed-DNA analysis in paleolimnology is currently rather active, and high-throughput sequencing has clearly facilitated the development of this research field. And you see in this graph the increased number of studies using metabarcoding and more recently shotgun sequencing in paleolimnology. These studies were mostly conducted in temperate and Arctic subarctic lakes, and they explore time scales from the century to million years time scale, so it's really diverse, with exciting innovative methods that are proposed to optimize the capture of ancient DNA, as for instance techniques based on the coupling of target capture technology with shotgun sequencing. One of the added value of this DNA approach is, of course, to expand the range of organisms that are detectable in sediment archives. And as illustrated here, the scientific articles published in this domain deal with various biological groups, including, of course, terrestrial taxa, but mostly aquatic taxa. And you see in particular a rather high number of studies focusing on phytoplankton groups, on cyanobacteria, on diatoms, and more globally on microbial eukaryotes. So I take the opportunity here to thank the SED-A-DNA Scientific Association. Among other activities, this scientific group organizes a literature survey. It is done under the impulse of young researchers, so I thank them, and it was extremely helpful to prepare this graph, to have the access to this information. And some scientists of this SED-A-DNA group have recently published an overview including methodological recommendation regarding the application of SED-A-DNA methods. So this review complements, I think, rather efficiently the existing papers and synthesis dealing with DNA-based methods in paleolimnology or publication dealing with all the specificities associated to the analysis of ancient DNA. So I do not go into the details here, but of course these specificities related to ancient DNA have to do with DNA preservation when aging in sediment, when burying in sediment, but also have to do with the risk of contamination by modern DNA, and more globally with all the precautions that are needed to calibrate or to authentify the results. So once again, I do not give precise detail, but you will find for sure literature on this topic. I'm shifting now to some illustration of applications of results we have had in our group, and we conducted studies to reconstruct the past dynamic of cyanobacterial communities and microeukaryotic communities, but I will focus on microeukaryotes here today. So just a short reminder regarding the large taxonomic and functional diversity of microbial eukaryotes. In this simplistic view of the pelagic food web, you see that you can find microbial eukaryotes within phytoplanktonic communities, but also within parasites, within bacterivores, within micropredators, or even saprophytes. And we applied a metabarcoding approach targeting 18S RNA genes. We performed, of course, replicates of PCR and replicates of sequencing for each sediment strata we analyzed in order to obtain in these inventories of diversity. From these inventories of diversity, we collected metrics on alpha diversity, beta diversity to differentiate species composition among periods, but we also considered the different trophic groups separately, mixotrophs, predators, parasites, etc. And we additionally, for some lakes, used this long-term data to construct ecological networks and to identify the co-occurrent taxa that constitute modules within the networks, and to identify also OTUs, which are important connectors or network hubs. All these metrics being, of course, mobilized to investigate the timing and magnitude of change, as well as the drivers explaining this rearrangement when it was possible. So I start the illustration of results with the case of Lake Bourget, a deep calcareous lake. So this lake is a good model due to its very clear and marked ecological trajectory, with a period of eutrophication in the 60s, 70s, 80s, and a progressive re-oligotrophication during which the relative importance of climate warming has certainly been reinforced, in particular since the mid-80s. So first, if we have a look to alpha diversity, for this lake at least, but this was not always the case for the other lakes we studied, but for these lakes, the Bayesian change point analysis we did could not reveal really marked temporal changes in the total richness of microbial eukaryotes or in the diversity indices we used, for instance, ill numbers or classical indices as Simpsons. However, considering the beta diversity and the composition of these assemblages, we could clearly show the rearrangement of diversity with a major change point in the mid-40s when eutrophication started, as it is revealed and illustrated here by the multivariate regression tree results. So this temporal shift in the 40s was also confirmed by other proxies and is also consistent with the global shift reported due to the great acceleration of anthropogenic pressure in the 40s, 50s. If we have, for the same lake with the same data, if we have a look now to the projection of dissimilarity indices on these NMDS, we see that even if the chemical quality of water has been restored, our data show that microeukaryotic diversity has not returned to its pre-eutrophication state. And this is obviously explained by the combined effects of different stressors, not only eutrophication. So we used the generalized additive models to explore the role of drivers, and this allowed us to verify that phosphorus was a major predictor of these changes of diversity, with a significant role in particular before the 90s. But we also detected that the positive anomalies of temperature were significant predictors of this rearrangement in the last decade. So this allowed to confirm that recent warming has curbed the real eutrophication process even for microbial taxa community. So if we compare the structure of this community at the beginning of the last century and in recent period, we see that the changes in beta diversity seems rather important. So we were of course interested to verify if we could see similar patterns or high magnitude of change in other lakes also. We therefore applied the same methodologies to a larger number of sediment cores originating from 51 different lakes, and we got some successful results for 48 of those lakes. And as you see here on the graph, these lakes are diverse in terms of altitude or regarding their current trophic stature. So here, for these 48 lakes, we applied a simplified approach consisting in the comparison of two periods, a recent period and a reference period taken at the end of the 19th century, meaning before the so-called great acceleration of anthropogenic impacts as it has been defined by Stephen and collaborators. From this larger dataset, we could observe that for most of the studied lake, significant changes were indeed detected in the beta diversity of microbial eukaryotes. But in particular, the highest magnitude of change were observed for all lakes located at low to moderate altitude. However, even for some high altitude lake, remote lakes, we could detect also significant rearrangement of diversity, as you see here in this graph, where all the dissimilarities between top and bottom layers are represented for the 48 lakes according to their altitudes. So the NMDS projection here with the same data give an overview of these modifications with past periods in pink and recent periods in blue. And this is interesting to notice this reduction of spatial diversity in recent periods. Well, this suggests, as it was previously reported for cyanobacteria, that a global homogenization trend might have occurred during the last century for these communities. And of course, this hypothesis needs to be further consolidated, but there are several other information in the bibliography suggesting this trend for the last century. A last illustration of those results Here we consider the composition and proportion of the different trophic groups, and the DE-Sec2 analysis suggests that mixotrophs and photosynthetic taxa have been favored by the recent conditions in the lakes, with in particular the increase of taxa within chrysophytes, dinophytes, and chlorophysian. So this is interesting to see that these data coming from DNA are consistent with other proxies as pigments, for which we have also quantified their quantity in the same sediment samples, or it's also consistent with few limnological data that were available for some of the studied lakes. So here we assume that the increase of these pigmented species we are detecting here is related to the global increase of nutrient over the last century, and also to the recent climatic conditions that have favored some species among these pigmented taxa. It was already reported for Lake Superior, where a steady increase of primary production was observed. But of course, the added value with this DNA result is to bring information about which taxa are involved in these processes. Let's come to the conclusion now. I have illustrated here in this talk only a very small part of the potentialities offered by DNA approaches. And the take-home message is probably that there are plenty of ecological issues that can be addressed using these DNA-based methods to complement limnological approach and to complement traditional proxies in paleolimnology. So it goes from the study of intraspecific diversity, it has been done in particular for diatoms, to the reconstruction of ecological networks. And you see here the example from the data obtained for Lake Bourget, where we see the changes in the proportion of modules constituting the network, and we could detected some OTUs involved as network hubs. So these DNA approaches could serve various ecological questions regarding the establishment of species, invasive species, pathogens, or questions related to the adaptation of population, to the presence of some functions that could be of interest. I have just taken here the example of toxin production, but there are a lot of other possibilities. And also some link with the dynamic of pollutants could be used to have some more paleo-ecotoxicological approaches that could be conducted. However, of course, as with any other new proxy, standardization, optimization, and calibration steps are essential. So these DNA tools are globally operational and can already give some very interesting results, but we clearly need to consolidate our protocols to perform more comparisons with traditional proxies, and also to integrate new methods that could help reaching the DNA signal from past taxa, since this DNA material is generally present in low quantity and is submitted to fragmentation and diagenetic processes. So to conclude, the coupling between neolimnology and paleolimnology will obviously bring added value in the next year by helping the characterization of pre-perturbation states and also for the development of models and scenarios of biodiversity. And of course, all these informations brings additional knowledge to explore the links between biodiversity and ecosystem functioning. So I want to thank you for listening to this talk, and to thank very warmly the numerous collaborators who were strongly involved in this work and the results I showed here. The list of the names is probably incomplete and I apologize for this, but I really wish to thank them, and in particular the young researchers who worked with me on this topic.